Thank you, PJ, and thank you to the organizers. It's a real privilege to kick off this meeting, so thank you. Hopefully I will do it justice. I'm Lisa Forbes Satter. I am a clinical immunologist, and so my rare disease space happens to be an immune-mediated disease. And so my colleagues and I have been trying to figure out how to do these innovative trials. And thanks to NCATS and the NIH a few years ago, they opened some funding and an opportunity for us to try this in what we call basket clinical trial. And so here's my steering committee. I just have to shout out that we're all jumping off the cliff together and having a great time. But without these colleagues, we wouldn't have been able to put this together. So I want to acknowledge their hard work, even though I'm the one standing up here. And so what we learned in the RFA from the NIH funding was that they wanted us to pick one drug to treat multiple diseases in one molecular pathway. And how do you figure out how to quantify efficacy for diseases that might share a particular molecular pathway but have nothing in common? And so of all of the kind of basket or umbrella trials that worked best for us, really what we thought was a basket-like approach would be the best. So not just, the same tumor type with different drugs, but mostly the same one drug for multiple diseases. And the advantages to that is that we can encourage partnership with pharma because we can collectively treat multiple patients with different diseases at once. We can try to facilitate this with smaller trials that might be more flexible, and then accomplish the ability to get some real data and some what we would consider regulatory efficacy data for us to be able to move on to indication for our patients. And so we chose a group of diseases in the JAK-STAT signaling pathway that are caused by activating mutations. And we were required to run the trial through an already established rare disease clinical network. And so the Primary Immune Deficiency Treatment Consortium allowed us to do that. It's a consortium that is in the RDCRN that has about 49 sites in North America. And they had already developed disease-specific organ modules to score disease activity. And the diseases that we were going to study were already under a protocol study. So it was the perfect construct for us to try to run this. And so what it allows us to do is, as opposed to a traditional clinical trial where we have to, cast our net wide for a lot of patients and look at a few endpoints, we can actually utilize this to see and treat a small number of patients, but really dig into the biology and go for a lot of endpoints and really be able to get not only mechanistic, but clinical efficacy data. So we proposed a basket clinical trial that shared the molecular etiology, and we had to describe the pathway, provide proof of concept that the drug would work, project, of course, put the trial through the RDCRN, and the Primary Immune Deficiency Treatment Consortium was that. And then, of course, we got NCATS funded this project. And so the key considerations while we set up this trial was that we were going to be able to do a lot of the immune profiling and mechanistic. Did the drug work? How did the drug work? How are we modulating? Could we learn about the biology? Because biomarkers aren't enough anymore for clinical efficacy and regulatory, but we wanted to be able to advance something that our clinicians could follow, write a clinical protocol, liaise with the FDA, develop relationships with the FDA that we could develop these protocols together. So we were accomplishing what we needed for our patients, but also we were devising a trial that we could use as a foundation for future clinical trials. Use these new modular CRFs that were based on disease scores from multiple diseases that we could adapt through heterogeneous disease phenotypes and quantify efficacy, and of course, make sure that our data was sound. So we have a basket clinical trial using a JAK1 selected JAK inhibitor to treat patients with JAK-STAT pathway disorders. if you look at this slide, these are the disorders that are under study for our trial, and you can see that the phenotype is completely different in each one of them. So to be able to actually quantify efficacy and understand whether or not we were going to be able to get the data we needed for indication, we had to come up with a system that we could compare varied clinical phenotypes together. So what we did is we went forward and we worked with the FDA to work on an IND. We had to evaluate the safety and efficacy and determine what the underlying defects were contributing and how the drug modulated that. And so our biggest challenge when we came to the FDA was control. We originally designed the trial that we would just look at clinical response based on the scoring. If you got the lowest score, you were doing better. If you got the highest score, the drug wasn't working, and we had a range in between. We originally set up the trial that each patient would be their own control, but the FDA didn't like that because they couldn't tell that if you took the drug away and gave it back, was it better? And so this whole idea of what the control would look like. In a rare disease population, sometimes these double-blinded, randomized control placebo trials just don't work because if we know that these drugs are good for the patients, it causes an ethical quandary for us as clinicians that we withhold the drug from our patient. So we had to come up with a way that we could assess regulatory efficacy without compromising care of the patient. And so what we did is we came up with a randomized withdrawal phase. So we had a dose escalation phase. So if you'll see 14-day screening phase, a 16-week dose escalation phase to the optimal tolerated dose. Then the patient would stay on that optimal tolerated dose for a minimum of 4 weeks before being eligible for the randomized withdrawal phase. Randomized withdrawal phase, the first sign of a flare, the patient could come out of the randomized withdrawal phase and go into the maintenance phase, because that would say that we could measure efficacy if we just showed that the patient flared. And then they'd stay in a maintenance phase and it would be a year-long trial. This was done in direct collaboration with the division at the FDA. We had several meetings. They were incredibly engaged because it was mutually beneficial. We got a trial design that helped with rare disease, where we could effectively measure multiple varied clinical phenotypes with a quantitative measure that could be compared across all of them. We got efficacy data, and then the FDA got a construct in which they could feel like we were measuring regulatory efficacy to put forth that in the future, if this was something that we could go for an orphan drug designation as such. So we had to define, here are our primary endpoints. Safety and efficacy obviously is important. Our efficacy measure was our scoring system. And so what we did is a score of 1 is basically asymptomatic, and a score of 5 is death. Obviously, hopefully none of our patients will achieve that score, but you had to enter the trial if your disease-specific score was a 2 or more, because we had to be able to show response down to a 1. And then they required us to define response. So that efficacy in the randomized withdrawal phase had to have a complete and partial response to enter it. And then we had to be able to modulate what the response was in the withdrawal phase and then after, when we put them back into the maintenance phase. So these definitions and that randomized withdrawal phase were very key for us. And then, of course, we have secondary endpoints that I think are important in most clinical trials. But the other really amazing piece of this is that it allows us to do system biology across the entire course of the trial. So not only are we developing a construct where we can take multiple diseases, treat them with one drug, develop clinical efficacy, regulatory efficacy, have the data that we can to go ahead and put towards indication, but we can also learn as much as we can from every patient that runs into the trial. So the important piece of the basket clinical trial in rare disease is that it's a good rationale. We really feel like the multi-omic approach and the partnership of the FDA was pivotal for us in being able to get an IND and move forward with this. We're really excited that if this approach works, that we are developing a construct by which we can take current drugs on the market, repurpose them for rare disease, and actually be able to do a relatively flexible and efficient trial to get our patients the drugs that they need. So with that, I have a lot of other people to thank, but looking forward to taking questions at the end and meeting all of you during the course of the conference. So thank you.
Thanks, Lisa. That was great. And next speaker will be Kevin Thornlow from Farming.
Great. Well, first of all, I want to thank NORD and the Symposium organizers for the opportunity to talk today. I think this is going to be a great meeting ahead of us. I'm very excited. I'm going to kind of continue the theme in the primary immunodeficiency space, following on from Dr. Sater, to talk to you about our efforts in developing a drug for common variable immunodeficiency. So common variable immunodeficiency is actually the most prevalent symptomatic primary immunodeficiency out of the very numerous numbers of different types of PIDs. And Dr. Sater spoke about specific monogenetic drivers of primary immunodeficiency, particularly in the STAT-JAK pathway. In the case of CVID, some of these patients do have genetic drivers that have been identified, but the majority of them do not. It's still considered that the disease is primary and that there's a genetic basis of the disease, but perhaps more complicated than a single gene. When you think about CVID, these patients can be divided into two different categories. Those that, due to their impaired antibody production, have recurrent infections, and that's solely or mainly the issues that they face. Those patients are pretty well treated, and we're very fortunate for this disease state to have good natural history data. You can see the graph on the right. This is mortality data out to 40 years, which is phenomenal in a rare disease. So we're very, very lucky to have that. Those patients do particularly well. The other 50% of patients have what's called complicated CVID, and I'll call that CVID-C. So they have the infectious issues, but on top of that, due to the dysregulation in their immune system, have a lot of different autoimmune and hyperinflammatory manifestations. And these are very complex. All sorts of different organ systems are affected. One patient may show certain symptoms, a different organ system may be affected, et cetera. So this is a very heterogeneous group of patients. There are no FDA-approved therapies for CVID-C. Typically, sort of broad off-label use of immunomodulators or immunosuppressives are used. And I show you this overall mortality curve on the right. A number of different manifestations have actually been associated with mortality. That includes splenomegaly, lung, and GI disease as well. So we've developed leniolisib for activated PI3K delta syndrome. And this is a specific monogenetic form of CVID-C. In APDS, those mutations cause a direct hyperactivation of the enzyme. Those mutations are directly in the enzyme. And leniolisib is a selective inhibitor of PI3K delta. So you could say sort of a perfect fit. So this was approved by FDA in 2023 for ages 12 and above, 70 mg BID dose. And you can just see some of the data here. Very nice efficacy responses in terms of reduction in lymphoproliferation in these patients. So why do we think leniolisib would be effective in this broader, more heterogeneous group of CVID-C patients? 3 points I really want to make here. One is the APDS experience. This is really proof of concept that PI3K activity can drive the disease, and also that leniolisib can benefit it. There are also other monogenetic forms of CVID-C that show increases in PI3K signaling. And the clinical characteristics, that basket of manifestations, is quite similar to APDS. But I think most importantly, going to sort of basic physiology et cetera. Point three is that PI3K delta is really important in lymphocytes in driving proliferation of those cells, has widely been implicated in its ability to drive autoimmune manifestations and inflammation in various organs. And these three sub-bullets here really encompass the heterogeneity and responses that we see in CVIDC patients. So what have we done at this point? So we've initiated a phase 2 clinical trial with leniolisib in CVIDC. Initially, we considered the potential to run a placebo-controlled study as a first study, phase 2 in this population. But sort of similar to what Dr. Sater explained, the rarity of this disorder combined with the severity of the disease and the limited information we had with leniolisib in this patient population, it was very, very easy to determine that that really wasn't a feasible approach. So we've really focused on going really deep in a small number of patients to rapidly generate some mechanistic data, safety, tolerability, and some clinical insights around efficacy. As I mentioned, the 70 mg BID dose is the approved dose in ages 12 and up in APDS, but it may not be the right dose in this patient population. So we've used an intra-patient dose escalation approach where each patient goes through the 10, 30, 70 mg regimen as shown here. We think this is really important due to the heterogeneity of the disorder. One patient may require a certain dose, whereas another one may require a different dose. So exploring that in individual patients we felt was really important. Now, we're not just broadly jumping into CVIDC, but we're requiring these patients to have lymphoproliferative disease. So this is going back to the benefits in APDS that I showed you previously. Patients need to have either large lymph nodes, large spleen, consistent with APDS. And we're not just interested in that, but we're interested in other manifestations as well because of the heterogeneity and unmet need here. So they need to have that plus one other manifestation. We're doing a very extensive 1-year retrospective medical history in these patients so we can compare before and after treatment. We felt that was really important to do individual patient comparisons, and we're going very deep in terms of biomarkers, et cetera. So how did we go about this? How did we really think about how to progress a CVIDC development program and initiate the work on this? And there's decades of experience, as I showed you, in this space. So it's really founded on the science and the medical understanding of the disease. We developed the trial in collaboration with leading global experts, including Dr. Sater, who sits here with us today. We're measuring what we consider to be clinically relevant, but also quantifiable endpoints. And that quantification, of course, can be difficult sometimes, but we feel like these things are quantifiable. And that data will then allow us to decide for future trials as we move into the more pivotal phase of development, how to prioritize these endpoints. As I mentioned, biomarkers, looking at lymphocyte subsets, which are really important and dysregulated in this disease, different cytokines and biomarkers to not only understand the patients coming into the study, but the impact that leniolisib has on those. We're also going to the extent of doing wider proteomic and transcriptomic work to look at changes in genes and proteins and signaling pathways. Because of the heterogeneity in disease, one patient may look somewhat different than another, and we want to understand this in detail. And this will all, obviously provide evidence, understanding the patient population, the benefits of leniolisib, and may, guide us to be able to perhaps enrich our population or focus it as we move forward. So I described to you that 20-patient trial previously. That's a phase 2 study in complicated CVID that's what we've termed gene agnostic. So some of these patients may have genetic drivers identified, but the majority do not. And we feel like this mechanism is relevant in both those groups. We're recruiting that study currently, but we're also recruiting a single center study in patients that have specific monogenetic drivers, a 12-patient study. And these are monogenetic drivers where there's evidence that there's increase in PI3K delta signaling in those patients. So we felt like this was the best probability of success in this heterogeneous population is to be able to initiate these 2 trials that will inform where we go next. So importantly, yes, we worked with world's experts, but we also have engaged regulators FDA, EMA as well, and patients as we've set this program up. And that's, as I think everybody in this room knows, very important in a rare disease setting, and particularly in a heterogeneous one where there's no prior drug development. So in terms of getting engagement and alignment on the rationale, mechanistically, unmet medical need in these patients, and the overall approach to development for this new medicine. And conversations with patients, very important, understanding what do they see as relevant needs. This is really important in heterogeneous diseases where one patient may have certain symptoms and issues and another one very different. So having those conversations with patients are important. We've also developed patient-reported outcome instruments to assess severity and impact. And we're piloting those, of course, in our 2 phase 2 studies. So with that, I will wrap up. I just want to thank the scientific medical community of today and previous that have done work in CVID. And I want to mention the 2 principal investigators for our phase 2 study, Gulbu Uzel at NIH and Dr. Jocelyn Farmer at Lahey Medical Center. And of course, I want to thank the patients, NORD, and all our regulatory partners as we move this new development program forward. Thank you.
Thank you, Kevin. Our next speaker is Scott Plotkin from Mass General. And Scott. Looks like those are Kiran's slides, so maybe we'll go to Kiran Musunuru next. Sorry, Scott. Unless you want to give Kiran's talk, but.
Like right on the presentation, we look at that.
You may get to the beginning of Kiran's slides, please.
Or the beginning of the 3rd talk.
Yeah.
Can you go to the beginning of the 3rd talk? That works good. Okay, it's gonna be one of those mornings, isn't it? Lovely. Okay, unexpectedly, I'm the 3rd speaker. I was sort of set up to the last one because I kind of diverge a little bit in the focus of my talk, but that's okay. You'll get it all in the end anyway. Well, it's a real pleasure and privilege to have the chance to speak to you today. I should know my relationships with a lot of companies with the word therapeutics in their names, but none of them are relevant to the subject of my presentation today. My lab and my collaborator, Becca Ahrens at the Children's Hospital of Philadelphia, CHOP, are working on a variety of metabolic diseases, so-called inborn errors of metabolism. One example is phenylketonuria, or PKU, well-known disease marked by very high levels of the amino acid phenylalanine in the blood, which, if inadequately treated, can cause a host of neurological problems, seizures, intellectual disability, and so forth. It's caused by a broken enzyme in the liver, and so the ideal treatment would be to fix the enzyme directly in the liver, and we can potentially do this with gene editing. Here are the most frequent pathogenic variants that cause PKU. The variants in red and gray are amenable to the gene editing technology known as adenine base editing, which can make A to G changes on either strand of a gene. The variants in light blue would need a different type of editor, such as a cytosine base editor or a prime editor. I'm going to focus on adenine base editing for today's purposes. The most frequent PKU variant that you see there in the list in red is the R408W variant, which accounts for an estimated 22% of variant alleles in the worldwide population of PKU patients. And we can potentially correct disease-causing variants in the liver, at least, using lipid nanoparticles, or LNPs, which are often compared to soap bubbles. Within the LNPs is a cargo, in this case, a messenger RNA that encodes a base editor protein, and a second, smaller RNA called a guide RNA that acts like a GPS and tells the editor protein where to go in the genome and where to make the corrective edit. And when these LNPs are infused intravenously directly into the bloodstream, they get taken up efficiently by the cells in the liver, the editor gets expressed, and then the editing occurs. So we made LNPs with an adenine base editor and a guide RNA whose GPS address directs the editor, points it to the site of the most frequent PKU variant. Using a mouse model that has PKU caused by the R408W variant, we found that the mice have elevated blood phenylalanine levels, just as we see in PKU patients. As you can see here with the orange and green points in the graph, healthy control mice in purple at the bottom of the graph all have normal levels. When we administered the LNPs to the mice indicated in orange, we found that by 48 hours, the phenylalanine levels had normalized, just 48 hours to correct a lifelong genetic condition. And this is a case where the biomarker phenylalanine is also the causal agent. So if you treat the phenylalanine, that's treating the disease. I'm not showing the results here, but we've observed that this effect lasts for the lifetimes of treated mice. So in principle, this treatment, the same exact treatment, should work just as well in human patients. Now here again is the list of the top PKU variants. We were able to pretty quickly figure out how to correct all the variants in red, 7 in total. This is still a work in progress, so we'll undoubtedly be adding more. But we can treat all of these 7 with the same exact adenine base editor. The only difference is the guide RNA, the address in the GPS. In other words, we can treat patients with any of these variants simply by taking the same LNP drug with the same base editor and changing one small part of the guide RNA. It's basically the same drug, just tweaked a bit to personalize to the patient who's being treated. So now there's the interesting question of whether we can go to a regulatory agency like the FDA and propose that they should let us do a single clinical trial with all of these variants, an umbrella clinical trial that enrolls PKU patients with any of the variants in red using slightly different versions of the same LNP drug. And we're going to be asking the FDA that very question. In fact, we have a pre-IND meeting with them next month to discuss this. Through work funded by the NIH's Somatic Cell Genome Editing Program, Becca and I hope we can submit a single IND application presenting a full set of IND-enabling studies for just PKU variant number 1, showing that the basic LNP drug is safe with toxicology studies in mice, non-human primates, and so forth. For the other variants, we'd simply submit cellular data showing that the editor and guide RNA correct the variant and don't have any concerning off-target editing, that is, unwanted editing of genes other than the gene that causes PKU. This would enormously streamline the development process compared to doing a full set of IND-enabling studies for each individual variant over and over again at great expense. And once approved, this IND would allow us to do the umbrella clinical trial. We could actually treat patients in a much more personalized way. But let's address a major issue, though, the serious potential for inequity of access to personalized gene editing therapies. As you saw, we now have base editing solutions for 7 of the most frequent PKU variants, and that's great if you're a patient who has one of those 7 variants. That covers more than half of PKU patients. But what about the more than 1,000 other variants that have been cataloged? What about those patients? And there's actually a bigger problem. Here's a figure from the most comprehensive study of PKU variants worldwide, plenty of data from some parts of the world like Europe, and then whole stretches of the globe from which there are little data, for example, the entire African continent, large parts of Asia. And this highlights that you can't make gene editing therapies for patients if you have no idea what genes and variants are causing their diseases. And as things stand, that's a big issue in many populations. And the upshot is that there's enormous potential for inequity here, a huge risk that drug development efforts are going to be biased towards certain genes and certain frequent variants in high-income countries. We're already seeing that happen, that there will be what we've come to think of as mutational discrimination. And our team has been thinking a lot about this problem and how we can make more accessible and inclusive gene editing treatments. Fundamentally, we need to shift from a disease-first or variant-first approach, which is what has dominated drug development until now, to a patient-first approach. So our goal now is to be able to rapidly develop and validate a corrective therapeutic for any variant in any patient with PKU, or really any other inborn error of metabolism that's caused by a problem in the liver. Each and every patient deserves a fair shot at this. So if we can get approval to go ahead with the umbrella clinical trial, the next step would be to work out a mechanism where we could efficiently add new variants to that umbrella. If we have patients with new PKU causing variants that we think we can correct with adenine base editing, we would quickly generate cellular data to document that, and then we would submit amendments to the IND, a process that we hope would only take a few weeks. And once an amendment is approved, then we can immediately make a personalized therapy and treat a patient. So let's now talk about a group of devastating ultra-rare inborn errors of metabolism, the urea cycle disorders and organic acidemias. They're centered in the liver, but the real harm happens to the brain. These are recessive diseases caused by pathogenic variants affecting any of the enzymes in the pathway shown here. They all result in high blood levels of ammonia and in some cases other toxic metabolites, which cause irreversible injury to the brain. The mortality rate in infancy is very high. I've been working with Becca Ahrens-Chop, who takes care of these patients, and Fyodor Ernov at the Innovative Genomics Institute at UC Berkeley to try to figure out how we can bring personalized gene editing treatments to these patients, just as we've been trying to do for PKU. Here's a list of neonatal onset urea cycle disorders and organic acidemias, along with various genes that can cause these diseases. From a regulatory perspective, this is more challenging than PKU because there are multiple diseases caused by multiple genes. They're far more rare than PKU. It's much more the case that patients have their own unique variants, so-called N-of-1 scenarios. We've been working out methods to be able to make personalized therapies for these patients as quickly as possible. Last August, the patient with the worst of the urea cycle disorders, CPS1 deficiency, was born. His name is KJ. I can say that openly because for better or for worse, he's just about the least anonymous baby in the world right now. A couple of weeks ago, we reported our effort to develop and treat KJ with a bespoke base editing therapy made just for him called KABE. This was published in the New England Journal of Medicine with an accompanying editorial written by Peter Marks. We were able to develop and manufacture the drug and have the FDA approve a single patient expanded access IND application in just 6 months. The FDA team were wonderful partners to us in this effort. KJ received multiple treatments at around 7 and 8 months of age. Although we didn't fully correct his CPS1 deficiency, I think, it does seem that we've turned a horribly devastating disease into a milder form of the disease. He's been thriving since we treated him. Now, you might think that this is a nice accomplishment, but paradoxically, I think this is actually not a great precedent for what we need to do going forward. It really isn't practical to do a single patient expanded access IND for every new patient. It's just not that scalable. Even though it will be a lot more work up front, we need to build a platform first as quickly as possible. And then we'll be in a position to very rapidly make personalized treatments for any number of individual patients with a much reduced need for regulatory interactions. So going forward, what we need to do is work with regulatory agencies like the FDA and establish that once we've done a full set of IND-enabling studies with a basic LNP drug for one variant, for one of these diseases, one of these genes, and obtain some clinical data in patients, we can then rapidly add new variants for that gene, either by having included them in the original IND or with simple protocol amendments supported by cellular studies only. And then we would propose that since all these diseases are closely related and have the same clinical endpoints, we should be able to add variants for any of the genes for any of the other diseases. It's basically the same drug. The only difference, again, is the guide RNA. It's the same target organ, the liver. We think of this as a master protocol for an umbrella of umbrella clinical trials. Now, after Becca made this figure, we actually thought about it more and argued a little bit. Is it actually an umbrella of umbrella, or is it actually a basket of umbrellas trials? And so we're sort of back and forth on that. But an umbrella of umbrella looks much more attractive than a basket of umbrellas, right? So we're sticking with umbrella for now. But eventually, this master protocol would be approved by a regulatory agency for broad clinical use. Then whenever a patient is born with one of these conditions, we find out the genetic variants, and then within weeks, we can design and manufacture the drug for the patient, and then we can immediately give that drug to the patient, and hopefully we're able to make a huge difference in their life. That's our vision. Let's see if we can make that a reality over the next few years. There are so many people who have contributed to this work, too many to go through verbally, but I especially want to highlight PJ Brooks, Tim Levote, and Chris Boshoff, who oversee the NIH's somatic cell genome editing program, without which we would not be able to do any of this work. And a very special thank you to KJ and his incredible parents, who have been amazing partners in this effort. Thank you.
Thank you, Kiran. And then finally, last but not least, Scott Plotkin from Harvard Medical School.
Wonderful. It's a real treat to be here today at this inaugural event. The program is outstanding, and I look forward to coming for many years in the future. Thank you to the NORD team for the invitation, particularly to Dr. Brooks and Dr. Nealon for inviting me to speak here about one of my passion projects, which of course is taking care of patients who have NF2-related schwannomatosis. So by way of introduction, I wanted to, we're a slightly different condition in which patients develop tumors. This condition is caused by pathogenic variants in the NF2 tumor suppressor gene. The prevalence is about 1 in 58,000, so not an ultra rare condition. And patients typically present the adult patients typically present with dysfunction of the eighth cranial nerve, that is hearing loss, tinnitus, and imbalance related to the presence of vestibular schwannomas, which are benign tumors that affect the eighth cranial nerve. Children, which represent about 20% of the patients who present to clinic, don't typically present with symptoms related to the eighth cranial nerve, instead present with symptoms related to meningiomas or visual problems or skin tumors. On the right, you can see the 4 different tumor types that occur in this setting. On the top left are the vestibular schwannomas I mentioned that are benign tumors affecting the eighth cranial nerve. On the top right are meningiomas, which are benign tumors that affect the lining of the brain. The left are bottom left is spinal ependymomas, which are gliomas, and the bottom right are non-vestibular schwannomas, because these schwannomas can occur in the brain, the spine, and in the periphery. And in oncology, which is my field, these are considered to be benign tumors and therefore not as significant as malignant tumors, but we know that the number and distribution of these tumors leads to a very malignant clinical course, and patients really suffer with the burden of their benign tumors. And so I want to start off by outlining from my own perspective, what are the clinical challenges in developing clinical trials for NF2-related schwannomatosis? Well, of course, the topic here is that it's a rare condition. One thing that we struggle with is the presence of multiple different tumor types in a given patient, as well as the presence of multiple tumors. And how do you study that complex issue? Although there is the temptation to think that there are hard endpoints in all of oncology, when it comes to these benign tumors, it's not like cancer where you can just look at overall survival. One thing we struggle with is how do we compare different results across multiple small trials? And then finally, how do we deal with the long delays between clinical trials as our patients desperately wait for new treatments? And so this led us to develop the Intuit NF2 clinical trial. I'm sorry that the print looks small from here. It is designed as an adaptive platform basket trial, and we've heard a lot about these designs with the previous speakers. And our goal is to rapidly and efficiently screen multiple drugs so we can nominate them for further study. And the big question we had to solve was about these multiple tumors. How do we address that? So we designed this as a basket trial because we wanted to include all 4 tumor types as target tumors, as tumors of interest, whereas previous studies had really only focused on vestibular schwannomas. And because patients have multiple tumors, we had difficulty dealing with it in a traditional oncology sense, so we decided to pursue a strategy in which we had a target tumor that was indicated by the treating physician. That was a tumor that was either growing most rapidly or was the most clinically significant. But in addition, in order to gather more information about the effectiveness of a drug, investigators could also nominate up to 5 non-target tumors. These were additional tumors that we observed for their response to drug, so that together we had both target and non-target tumors that we were going to follow. And the goal here was to really maximize the amount of information that each patient contributed, because in the past we had been looking at single tumors, and in fact we're not capturing the totality of the effect of these drug treatments. And of course, the platform trial, as we've heard before, is our way of bringing multiple drugs to bear as quickly as possible. And in that setting, as we'll talk about, the design allows you to rapidly add new drugs for study. And then finally, we had long discussions about whether to have a placebo control. We talked for many years with our patient community and ultimately decided not to pursue a placebo control in this design and instead to look at natural history based on feedback of our community. And so this is the design of the Intuit NF2 trial. In the top left, you can see the top box is the master protocol, which is the protocol that allows for intake and distribution of patients. In the master study, any patient with NF2-related schwannomatosis who has a progressive tumor of any type then gets placed into one of the tumor baskets based on their target tumor. And they then get randomized into individual drug sub-studies, which is the box at the bottom. I don't think I have a pointer, so I apologize. We currently have 2 drugs under study. Drug sub-study A is with a drug called brigatinib. Drug sub-study B is a drug called neratinib. And patients are treated with these drugs until the time that the target tumor grows. At that point, patients come off of study and then get resent back into the master study so that they can be re-randomized to another drug as quickly as possible. And we've now had a number of individuals who have been treated rapidly with 2 drugs in a relatively short period of time. You can see on the right the 7 participating institutions. I want to recognize this huge collaborative effort and all the site PIs who are helping us with the project. So as I talked about, we focus on having a master protocol and drug sub-studies. In the master protocol, we're enrolling up to 80 participants. And just to reiterate, the master protocol defines the rules of the study. There are no drugs. It's in fact an allocation protocol as well as a way of defining what common response criteria are. I've listed below on the left the key inclusion and exclusion criteria. I won't go through them because this is just one specific rare disease, but to point out that this is defining the patient population that is in need of a new study drug as opposed to other standard treatments. On the right is a description of the sub-studies, and here you can see we enroll up to 40 eligible patients for each study. And we require that each basket is filled at least partially. So each basket must have at least 2 participants. And as I mentioned, we follow both target and non-target tumors, and we hope that these trials will last approximately 2 to 3 years. Our primary endpoint is tumor shrinkage, and so that way we are lucky, quote unquote, to have MRI scans, but we know that we'll never get approval for a drug based on tumor shrinkage because that's not a clinically meaningful endpoint. Instead, we have a number of important secondary endpoints that include other things such as hearing, pain, and quality of life issues that supplement our primary endpoint. Here's an example of how we do our adaptive design. I mentioned that our first drug is called brigatinib. It is an FDA-approved drug for treatment of lung cancer. It's very favorable, an oral drug given once daily with a safe safety profile. In the top right, you can see in stage 1, we enroll 20 participants with 2 participants per basket. We then perform an interim analysis. If no target tumors shrink, we terminate the trial for futility. So we do not want to enroll patients into a drug that we believe will not be active. However, if there's at least one tumor that responds, we then proceed to stage 2, where we enroll 20 additional participants. And what's important here is that in the interim analysis, we try to determine which 2 baskets are most promising, and then we enroll people only into the 2 most promising baskets. The goal is to allocate patients away from futile drugs and into the best sub-study, best target tumors that are available. And so I wanted to just mention very quickly, funding is such an important issue, I think, in all of our communities. Our goal was to really leverage public-private partnerships as a way of managing the cost here. And so I really want to highlight the critical role of the Children's Tumor Foundation, who brought together a lot of academic centers to do the drug screening leading up to this and to run the clinical trial. And the goal, of course, was to use this infrastructure to lower costs. The sources of our funding are listed there, again, showing the Children's Tumor Foundation as well as NCATS, which helped us with our drug screen. We've worked closely with the National Comprehensive Cancer Network, Takeda, and Puma Biotherapeutics, and the goal is to really move these studies forward rapidly. I'm running out of time, so I'll just quickly say that for agent selection, our goal has been to repurpose drugs and not develop new drugs at this stage, although, of course, we want to develop drugs that are very specific. And the way we've done that is to really leverage our preclinical laboratories to find efficacy of drugs, of established drugs in NF2 deficient tumors, And then to make sure that we felt that their safety profile was suitable for a rare disease population and with a focus on pediatric patients. We don't want to leave behind any of our pediatric patients, and brigatinib fulfilled that. And my last slide, just to talk about the key issue of access. If you find a repurposed drug that works, how do you then make it available to your patients? And so I'll just briefly say that our goal has been to work with the National Comprehensive Cancer Network. They have a clinical practice guidelines in oncology that describe best practices and provide evidence that can be used to explain to insurance companies why a repurposed drug that is not approved for your condition might be a reasonable option. And with that, I also want to thank a huge group of team and to thank you for listening today.
Thanks, Scott, and all the speakers. I would really like to give audience members an opportunity to ask any questions that you may have. I'd like to do that. While we're waiting for people to come up to the microphone, though, let me start with a question for Lisa. If you could talk a little bit about your experience in working with pharmaceutical companies and doing these kind of novel clinical trial designs.
Thank you. Partnerships, I think, has been a key theme throughout many of our talks. And so one of the key partnerships is, of course, especially for the repurposing of drugs that, Scott and I particularly do, and that farming is moving towards, is an important partnership. So for our trial, we partnered with a particular pharmaceutical partner and have really struggled with what we didn't think we would struggle with. So we thought funding would be an issue. When we started this, we weren't quite sure what our relationships and our discussions with the FDA would be like, which we have been fortunate in to have a partner like NCATS and PJ, and that our funding is intact, and the FDA has been phenomenal in helping us really drive this construct. And our industry partner decided that they were going to close the drug program that they had provided for us. So in the last year, instead of starting our trial, we have been negotiating with them alternative ways to use other products, other similar JAK inhibitors, and then expanding our footprint to engage other industry partners that have similar drugs that would be as efficacious in our patients. As we do use these drugs off-label for our patients, we have developed other partnerships, and this is a team sport. So navigating how to do that, how to maintain this really important trial contract that we've developed and acquired drug has been another challenge. So when we started this journey 5 years ago, we thought that the drug was the easiest thing because we had a partner that was excited to work with us, and that all of the other hurdles that we would encounter would be our struggles. But it turns out that competing priorities are important, and leveraging multiple partnerships and diversifying your ability to understand what the landscape looks like has been a really important lesson for us. So I have cautious optimism that by the end of the summer, we will have secured our new drug for the trial that will be just hopefully as efficacious in our construct, but that's kind of our current hurdle.
Thanks, Lisa. I'll go to some questions. If you could briefly introduce yourself.
Hi, Chris Vlahos, Acadia Pharmaceuticals. My question is for Kiran. Very impressive data. I'm curious if your approach will work in tissues aside from the liver. For example, skeletal muscle for dystrophies, maybe even across the blood-brain barrier.
Yeah, so a great question. So it all comes down to delivery, right? So the reason we can readily do liver is because the liver's role is to clean things out of the blood. So almost anything you put in the blood will get taken up by the liver. And so that's been hugely advantageous in developing these gene editing therapies and deploying them to take on diseases that are centered in the liver, like inborn errors of metabolism and a variety of other diseases. And that's been where, with respect to somatic therapies, the most progress has been. the overall approach I articulated should be just as valid regardless of the target organ or the set of diseases. It just requires that we get to the point where we can reliably and fairly specifically and efficiently to deliver to that target organ. So for example, you mentioned muscles. The only credible way we have right now of delivering a large cargo like, a gene encoding an editor and a guide RNA and so forth would be a viral vector like AAV. It has its pluses and minuses, and I would argue when combining with gene editing, probably more minuses than pluses. At least that's in adult patients. That is not to say that we won't figure out collectively as a field going forward non-viral methods like lipid nanoparticles that we can somehow engineer them either chemically or by attaching moieties to somehow be able to get them to efficiently transduce muscle cells and satellite cells and the cells you would actually want to target. It's a little bit of a tangent, but one thing that we have found in my laboratory and my collaborators at CHOP is that if you take standard lipid nanoparticles, nothing special about them, the same ones we're using to target the liver postnatally, and give them prenatally, and I'm not talking about human beings, obviously, but I'm talking about mice, sheep, monkeys, what we have found is they actually transduce the muscles very effectively. We can actually get very high level, maybe not very high, but high level base editing in these models, which suggests that if you really are interested in targeting the muscle, the best possible window might actually be prenatally. And for certain diseases, that might make sense, congenital, muscular dystrophies or congenital heart disease or so forth. But, I think there will be a lot of progress on this front. We're already seeing at least the preclinical level progress in being able to deliver, say, with lipid nanoparticles to hematopoietic stem cells in vivo and to the lungs in vivo. So I think all these things will come in the fullness of time. And once it is possible to target an organ, then I think the platform approach will be just as viable there as it is for liver-centered diseases.
Thank you.
Right. Yeah, and I might just say as part of the CG program, we actually are funding a prize competition for better ways to deliver genome editors to different parts of the body and across the blood-brain barrier. Next question.
Marissa Crayer, Chief Medical Officer at Alterna. So thank you so much for this. One question is actually getting to these clinical trials. And so as we think about basket trials and different diseases, the agencies ask for animal models in IND-enabling studies to be able to get to the various diseases we want to include in basket trials. I wonder about your different experiences and input from the agency as well, and thinking more about in vitro.
I can start with that. So for the group of disorders that we had, we were fortunate enough that we were going to a pharma with a pharma partner that had already had the drug in study for other indications and had done all of those. So the repurposing for at least our field and for identifying technologies that are already out there was a key driver for us because we had clinical efficacy for the class, but then our industry partner had already had the drug under study, had a dossier with the FDA. And so we were able to leverage that with our cross-reference and that such. So that made it facilitated a lot easier. I think your question really is when there's a new molecule. And so I think that the diversity of partnerships, you'll hear from some other colleagues later this afternoon that have taken a new molecule and have to do that as well. So I'm going to call you out, Manish. So if you want to look for Manish Butte, he might have some answers to that question. But I think that that's really the paradigm is that our FDA interactions were already past that because of the data that existed.
Kiran, do you want to address that one?
Yeah, absolutely. So I have a very different perspective because I'm using gene editing, which offers a very unusual opportunity. So I'm going to say something a little bit provocative. So CRISPR, gene editors, don't discriminate. They get at the root molecular cause of disease. They go wherever you point the editor, right? It will go to wherever you say to go. Gene A, B, C, variant X, Y, Z. It doesn't care. I'm anthropomorphizing, but it doesn't care. It's going to go and it's going to do its thing, right? And so the distinctions we place on diseases are less relevant when you're actually at the molecular root cause. And so the argument I'm going to advance here, and I have no idea if the FDA will be open to it, but eventually we will be approaching them with this kind of argument. So forewarned, is that if you're thinking about biological mechanism, then it's almost tautological that if you have a disease-causing variant, you can target it in the specific organ, say the liver, and you correct it either partially or hopefully more fully to wild type, then per force, you will be improving the disease, right? And it doesn't really matter what the mechanism of disease is. It doesn't, it doesn't matter the mechanism, it doesn't matter the disease, it doesn't matter if it's XYZ, it doesn't matter if it's only affecting the liver, if you're targeting there, or if it's a systemic condition that's caused by a fault that can be localized to the liver. It doesn't matter. You're treating at the genetic level. You know how the drug works. You know how it targets the organ. You know how it makes the change. The rest doesn't matter. So talking about umbrellas, ideally, you expand the umbrella until it contains all diseases that are centered in liver, where you can have a therapeutic benefit by making a corrective edit there. That is the end game. That is where we need to get to. So then it doesn't matter if you have, a very common disease or a rare disease or a disease that's so unusual that it doesn't even have a name and you can't even really fully understand or define it. As long as you know the genetic cause, you can address it.
Totally
Thanks.
behind you.
Katie's down here waving me and saying we have to end up now. So thanks very much. I'm sure all the speakers will be available to answer questions perhaps at the break. And thank you very much.